Hello dear, today the discussion topic is biotechnology. Actually this topic under this biotechnology today we will discuss its unit 5 which contains fermentations which is comes under 6th sem biotechnology syllabus as well as syllabus in PCI curriculums. Okay, then under this we will discuss fermentation technology and biotechnological product, fermentation methods and general requirements, study of media, equipment, sterilization method, etc. Comes to first point of our fermentations. The science of fermentation is called zymology. Then the fermentation means any process that converts larger molecules into smaller and simpler molecules using microorganism. That means microorganism plays a vital role in fermentations. Generally this fermentation process converts larger molecule to simple and smaller molecules which is useful for pharmaceuticals and as well as for food industry. Okay, let's simplify it. It's a metabolic anaerobic process that converts sugar means it is a large molecule to acid, gases, alcohol or any metabolic byproduct. So thus in biotechnology fermentations is a process since which microorganisms are grown in large quantity by providing them favorable conditions which converts organic complex substances to simpler compounds. That means without the help of microorganism most probably the fermentation process cannot be fulfilled. Microorganism plays a vital role in fermentation process. Fermentation also occurs in oxygen starved muscle cells as in case of lactic acid fermentation, and this is the step. Okay, you are seeing in the screens, this is the steps. Then comes to objectives of fermentation process. The major objectives of fermentations are to produce large amount of microbial cells or biomass, large microbial colonies, to produce microbial enzymes, protease and invertase enzymes, just like to produce microbial metabolites of glucose and lactic acid ethanol. See, question gote manku asibo je why we are go for microbial metabolites, because these microbial metabolites are very vital for pharmaceutical use. Means many pharmaceutical important materials they are prepared by the fermentation process in which the microorganism plays a vital role to produce recombinant products. Example, production in several vaccines eggs. Fermentation is a vital process for recombinant products, DNA recombinant technology or other technology in which vaccines are produced. So I am outlining the objectives again: to produce microbial biomass, to produce microbial metabolites, to produce microbial enzymes, to produce recombinant products. Then come to the next point: basic steps of fermentation process and general requirements. What are the basic steps? Steps are simplified as follows. You are observing in slides, just see the basic steps of fermentation. Number 1, formulations of fermentation media. It is chemically a cell culture media which is a mixture of molecules used for cellular metabolism and growth. Example, carbon sources, energy sources, oxygen and nitrogen. Means a media is very much necessary, because in a suitable media microorganism can grow. Each microorganism they specify a suitable media. Means for a particular microorganism a particular suitable media is necessary. Without the media they cannot grow and they cannot participate in fermentation process. Then the next step is sterilization. It is very vital. Sterilizations of fermentation media, that means except our desirable microorganism there should not be growth of other microorganism. Other microorganism बढ़ी पारिबे नहीं, केवल सही माइक्रोऑर्गेनिज्म माने बढ़िबे जो माने फर्मेंटेशन प्रोसेस को कैरी ऑन करिवे. सेथिपाई दी सिस्टम शुड बी कंप्लीटली स्टेरिलाइज्ड. देन inoculum formulations, productions of an active pure culture in sufficient quantity to inoculate into the productive vessels. Then fermentation, growth of the microorganism in the production fermentator under optimum condition for formation of byproduct. When the fermentation process is going on, a suitable media which help to grow of microorganism, and that microorganism carry on the process of fermentation. After completion of fermentation, the most important step is extraction and purification. Means what product we desire, that should be extracted and that should be purified. Impurity should be discarded and pure form should be adopted or collected. This is the steps, or this is the flow chart you can see in the screen. Just see, which already we have discussed. Just see the flow chart. Then come to next step, that is basic steps in the process of general requirements. The general requirements of fermentation process are: number 1, fermentation medium, which we have discussed, inoculum, fermentator, sterilization, growth of organism and extractions. Already we have discussed in the last slide. The last step is purification and filtration. Purification is very much necessary and extraction is very much necessary. Impurity should be discarded. Then comes to the method of fermentation process. Okay, what are the main important method adopted in pharmaceutical industry and food industry for the process of fermentation? You can see here batch fermentation process, then 3 method of fermentation process. Second one is continuous fermentation process, then solid state fermentation process. I am outlining the 3: batch fermentation, continuous fermentation and solid state fermentations. Comes to the next point, fermentation medium, medium with nutritional and hormonal requirements of cells in optimum concentrations. That means when we are choosing a media and prepare a media, the media should be contains nutritional substance which help for bacterial growth and hormone which accelerate the growth process. Then small modifications in the medium could change cells instability, product quality and yield. That means all parameter should be constant and continuously monitored. Then compositions of the media, I am giving very shortcut presentations of compositions: carbon source, nitrogen source, inorganic salt, vegetable and animal fat, growth regulators, trace elements, buffer to maintain pH, oxygen. All these are important, out of which vitamin B12 plays a vital role as growth regulator, and different trace element like zinc, copper and sometimes selenium are also used. Then buffer is very much important. All of you know, in any kind of pharmaceutical, okay, when buffer is used, which aims is to maintain pH. Then inoculum, it is the pure bacterial cell culture that is introduced to the medium. The cell then grow in the medium conducting metabolism. You can see here the equipment. This equipment is known as fermentator. The various parts we will discuss. First you go to fermentator. Fermentator ta kon ame janiba? A fermentator is a device that accomplishes the fermentation process by the help of certain microorganism, and thus it is also referred as biofermented fermenter or bioreactor. Kahinki a naata biofermenter or bioreactor? Because it carry the biological process. Biological process means substances like antibiotic, enzyme, beverages. Beverages means for food industry, etc. It is helpful. See the different parts of a simple fermentator. It is the schematic diagram of a fermentator. Then come to next part. I will enlighten you the different equipment in its purpose of the fermentator, which we have observed in the last slide. First is top plate. The main importance of top plate is very simple, it cover. Then Clamp. Clamp, what is its role? Top plate compressed into vessels using clamps. Then another part is seal. What is its role? Separates top plate from vessels. Then another is vessels. क्या है vessels? Glass jacketed steel with pots for various outputs. Then driver motor, driver shaft, marine impeller, waffles, flanger, exhaust gas cooler, inoculation needle, then feed pump, then peristaltic pump, then series pump, then exhaust gas analyzer, then sample piece, then three-way inlet. These are the various important parts of the equipment or fermentator. Then let's come to the descriptive part of equipments. Specific parts of fermentators are: number 1, body, okay, which is made up of glass. Advantages: given smooth surface, non-toxic, corrosion proof. Then seal, to seal stop plates and vessels to prevent air leakage. Types of seals: gasket seal, lip seal, O-ring seals. Then this is the equipment, the different agitator impeller mounted to a shaft. You can see it in the slides. Then here is the equipments. The four part, that is waffles. These are normally 4 waffles incorporated into a vessels. Then spazzers, the air introduced device in the fermentator is the spazzer. You can see it in the slides. Then large scale productions of fermentator design and its various controls, controlling devices for environmental factor. In any microbial fermentation, it is necessary not only to measure growth and product formation, but also to control the process by altering environmental parameters as the process of proceeds. Then hazard assessment symptoms. A hazard assessment is the process used to identify, assess and control workplace hazard and the risk of worker health and safety. It is very necessary because safety is very necessary for the product as well as for the fermentation process as well as for the workers. Okay. Here we will discuss large scale production or fermentator design and its various controls. Uses of computer in the controlling process of fermentation. We know nowadays computer is the vital parameter for any kind of processing, because modern softwares or developed softwares are there which can automated controls a lot of process. Okay. So computer technology has produced a remarkable impact in fermentation work in recent year, and the computer are used to model fermentation process in industrial fermentators. Okay. So come to the next point, citric acid productions. Okay. All of you know citric acid is naturally available in citrus fruits. Citrus fruit कहला माने आम मन को आसीबो orange and lemon. Exactly, you are right. These are the natural source of citric acid. Okay. Here we will discuss different applications, uses, industrial productions and large productions of citric acids. Okay. Citric acid is a usual occurring acid found primarily in several varieties of fruits and vegetables, with citrus fruits such as lemons and lime containing the highest amount of citric acid. This organic acid has many uses, including as a food, additive, preservative, ingredient in cosmetic products and a powerful cleaving agent. And also citric acid is believed to be participate in immunofunctions. Citric acid is the most important organic acid produced and existing in use of food and pharmaceutical industry. Citric acid is a weak organic acid found in citrus fruit, particularly in lemon and orange. It is good natural preservative and is also to add an acidic taste to food and soft drink. So again I am having a very good chart which demonstrate what is citric acid. See, natural intermediate in crop cycles, produced in anhydrous or monohydrate forms, produced by several modes and bacteria forms a variety of substitutes. See the beautiful schematic diagram. Then history. The discovery of citric acid has been credited to the 18th century Muslim alchemist Jabir Ibran. Citric acid was first isolated in 1784 by a Swedish scientist or chemist. See, this is the structure of citric acid. This is the chemical structure of citric acid. Then come to microorganism used for citric acid production. Large number of microorganisms including bacteria, fungi and yeast have been employed to produce citric acid. The main advantages of using this microorganism are: it is easy for handling, its ability for fermentation, a variety of cheap raw material, that is very important, it is economical, high yield. Microorganism which are participate in citric acid formation. First come to fungi, different species of fungi are there. The most important species are Aspergillus niger and other below mentioned fungi. The most important bacteria participate in citric acid formation is Bacillus licheniformis and other below mentioned bacteria. See, the most important yeast species are Candida topicalis and other below mentioned yeast. Then comes to citric acid production by three process: surface fermentation, submerged fermentation, solid state fermentation. What is surface fermentation? Let's come to know. Surface fermentations using Aspergillus niger may be done on rice bran, as is the case in Japan, or in liquid solution in fat albumin or stainless steel part. Then submerged fermentation. What is it? Let's see. In this case, the strains are inoculated of about 15 centimeter depth in fermentation tank. The culture is enhanced by giving aeration using air bubbles. Then it is allowed to grow for about 5 to 14 days at 27 to 33 degrees Celsius. Then the citric acid produced in the fermentation tank and it is purified. Then come to solid state fermentation. It is the simplest method for citric acid production. Solid state fermentation is also known as Cups process, was first developed in Japan. Then separations, the biomass is separated by filtration, the liquid is transferred to recovery process, separation of citric acid from the liquid preparation, calcium hydroxide is added to obtain calcium citrate. Then comes to factor affecting citric acid production. Number 1, nitrogen sources. Number 2, pH. Number 3, aeration. Number 3, trace element. Number 5, temperature. Then industrial productions of citric acid, about 99% of the world production leading to microbial process, surface of submerged cultures. 20% used in pharmaceutical industry as antioxidant, to preservative purposes and to effervescent purposes, pH corrections, blood preservative, or in the form of iron citrate, tablet, ointment and cosmetic preparation. Chemical industry remaining 10% softening and treatment of textiles, also used in the detergent industry as a phosphate substitute. Then application used in citric acid, food and drink, the most important role in food industry, preservative and flavoring agent, emulsifying agent in ice cream, household cleaners, kitchen bathroom sprayer, cosmetic, shampoo, body washes, wash cleaners, nail polish, hand soap products. So, you can conclude there is vast use of citric acid in pharmaceutical as well as food industry. To cure kidney disorder, what is the role of citric acid? Sodium citrate, acetic acid is used to prevent kidney stones. What are the side effects? Taking excess of citric acetate in combinations with sodium citrate may lead to kidney failure. Taking citric acid with empty stomach may lead to stomach or intestine side effect. So, it is better to take citric acid preparation with mild food, and it is better not to use excessive citric acid because it lead to kidney problems. It may also lead to muscle twitching or cramping if used in excess amount. It can also cause weight gain, swelling, fast heart rate, slow or rapid when it is used beyond the large dose. Largest production in the world. Here I have prepared a slice. USA. From which source? Grapefruit. China. From which source? Mandarin. Brazil from sweet orange. India from acid lime. Italy from lemon. So, here we have discussed the various facts and fundamentals of fermentation as well as the processing of citric acids, its pharmaceutical importance and its food industry importance. The slides I think is beneficial for Pharm 6th sem student for the subject of biotechnology. Thanks.